I think Kate is going to blow your minds and prove you wrong, haters. So she's going to bring it to you, and you better bring your questions. Bring them hard. And she's going to tell you a little bit about her work first, and then she's going to take the questions that you have. So, Kate, if you want to maybe give a little intro into who you are, and then talk about your work a little bit, and then we'll take questions from the audience.
Awesome. So hi, everyone. Can you hear me okay? I'm only using my inbuilt laptop microphone, but it should be fine. So what I do is try to make life from scratch. And while it kind of sounds crazy, it's not as crazy as you would think. Basically, the goal is to not make a Frankenstein monster or anything like that. The goal is to make a living cell that comes from non-living components. And this, when we think of it as basically ultimate biological engineering. We're trying to build living systems from well-defined non-living components. And the reason this is cool for me and for everyone else who works on it is when you work with live cells, a lot of the time you ended up scratching your head trying to figure out what the hell happened in your experiments, because live cells don't always, most of the time, they don't do what you want them to do. And when that happens, you have no idea what went wrong. Your culture didn't grow, or the protein you're trying to express for some reason didn't express. The pathway you're studying ended up not behaving like you think. And the reason for that is because live cells have their own things to do in life. They have their own endogenous pathways that are too complex for us at the moment to fully understand. With synthetic cells, the hope is that we can build the systems from bottom up, basically put together living systems from specific components, and then make this system more complicated by either evolving it or adding one piece at a time in a way that's completely controllable and understandable. So at the end, we will have a living system that is engineered in the lab and is completely understandable. So basically, the motto of our field is that famous quote from Richard Feynman. I know Josiah's laughing. Everybody always quotes that that when they talk about building life, and I will too, because otherwise it doesn't count. What I cannot build, I cannot understand. And that is really true about what we're trying to do. We cannot, as biologists, cannot claim that we understand life unless we actually can build it from scratch. And that brings up another problem. Biology is terrible at reproducibility. And when you try to figure out why, you come to the conclusion that the sample size of all modern biology equals 1, because we only know one type of modern terrestrial biochemistry. There's only one type of life that we know of, and that all of the people that ever studied biology studied this one type of life. You think there's a difference, you're studying a bird, or you're studying a bacteria, or you're studying some weird slime mold, but it's all the same biochemistry on the fundamental level. So we don't actually have anything to compare to. So if you observe some process that's universal to all known domains of life, you don't know if that process is unique to life as we know it, or it's some elemental process necessary for the broad phenomena of life. And so for that reason, we're trying to make, I don't want to say different life forms, but different instances of biological processes that people can agree are life. And then we can compare what we're seeing in our terrestrial biochemistry to some other kind of life processes and figure out what's actually unique versus what's kind of specific to our own life forms. And that has some cool applications. For example, that's just kind of a quick, relatively useless at the moment, but really interesting application is if we go out and find some organics on Mars or under ice on Europa, how will we ever know if it's life and if it has ever been alive? If we can only define life as the one thing we know on Earth. We need to make more kinds of life to be able to truly understand what biological life is, and if we find weird life elsewhere, for how to recognize it and what to do with it, kind of how to study it. So that's a kind of a far out there application that makes me really excited. The less exciting but the applications that actually pay the bills of our field right now are the biomedical applications. So a lot of you probably know about cell therapy and how CAR T cells and other modified cells and modified bacteria are kind of a hot thing right now. They're hot, but they're also kind of annoying to work with because a lot of, again, cells have their own business to do. And if you reprogram them to do something, for example, target cancer cells, they will go do it if you're lucky, but they will also go do other stuff like screw up your immune system. And if you build a synthetic cell for the same purpose to go after a specific cancer drug, cancer cell, for example, that synthetic cell, because you built it and you programmed it, will not go on and do something else. And this is not as crazy as it sounds. We actually, as of last year, we can say this has been done in mice. We can show that you can take a synthetic cell, put it in, not single synthetic cell, but you can take a tube of them, put them in mice, and if you program them to target a specific solid tumor, they actually go and do their thing and the tumor shrinks. Obviously, what works in mice doesn't always work in humans, but that is kind of where the field is right now. We know this technology is at least viable enough to keep trying. Another reason that makes me personally excited about this technology is in biohacker labs, you don't always want to do BSL-2 experiments. In fact, you probably shouldn't be doing BSL-2 experiments. But there is a lot of interesting stuff that you can study, like human pathways, human viruses, human pathogens, that you cannot study without BSL-2. But if you use a synthetic cell model, for example, that expresses a receptor on the surface of a synthetic cell that a certain virus can bud out from, then that is not BSL-2, because that is essentially a non-living, as of now yet, system. So this is kind of a cool tool that can be used to study things with the level of complexity that still makes sense, but lower than you would need to get an approval for anything above BSL-1 for your lab. So now, kind of something I probably should have started with, which is what actually is a synthetic cell. So that's a very good question, and nobody knows. This field is young enough that everyone working on it kind of have their own definition. We mostly agree that a synthetic cell has to resemble a natural biological cell in gross morphology. So we need a compartment that encapsulates a protein expression system. So you need to have genes that are then transcribed and translated inside certain compartment. The identity of that compartment differs depending on who you talk to. Most people want to do it in liposomes because lipid membranes is what natural cells use. So if you make an artificial lipid membrane, that's called a liposome. And so it's basically a soap bubble. You take lipids that form spontaneously at the right pH, a bilayer membrane that resembles, to some approximation, a natural biological membrane. So that's what my lab does, and that's what pretty much most of the field does. But there are also people doing really cool work using either droplets or either oil droplets or coacervates as their compartments. Basically, any way of keeping crap together is a compartment, and we're happy with that. It could be a liposome, a lipid vesicle, like my lab is using, or it could be a coacervate, or it could be a oil droplet, like in microfluidic systems. Any way of concentrating and keeping stuff together. So that's a compartment. And then the real interesting stuff goes on inside, which is protein expression system and genes that you actually put in there. So when we start building a synthetic cell, there are no genes in there. There are no genes coming naturally from the cell extract or from any other component we're using it. We put every single piece of DNA in synthetic cells. So that gives us the flexibility to program what kind of genetic circuits we want to have in those cells. That also brings a lot of problems because we don't have as fine control over gene expression. As you probably know, cells spend a lot of time and money on controlling timing and levels of gene expression. That's basically the whole thing about how cells regulate their life cycle, how cells regulate division process, is by regulating when each gene gets expressed and at what level. And there's a lot of complex machinery doing it. We don't have that machinery in synthetic cells, which limits our designs to relatively simple genetic circuits that can be regulated either by small molecules or proteins. proteins like transcriptional activators or recombinases. And so that's kind of the main thing that my lab is doing right now, is to build complexity of those genetic circuits inside synthetic cell. We want to be able to have our genetic circuits expressing metabolism, the synthetic cell metabolism, that's as complex as possible without completely losing track of what are we expressing. So we don't want to end up with a situation where we use 50 genes and we don't know what we did.
Okay, has anybody ever just like taken a genome or genes and put them in a synthetic cell like you're just talking about?
Yeah, people did, and it didn't work. People took an E. coli genome and put it in E. coli extract. Nothing happened. What we're trying to do right now is, that's actually a project that we're working with in collaboration with the Venter Institute, is we're trying to take genome of the, so Venter Institute built the smallest known living cell. It's called SYN 3.0. They call it synthetic cell. I disagree with calling it synthetic because it's made from living cells. It's made by simplifying known living cells. But anyway, it's the smallest known organism. It's based on a bacteria called Mycoplasma, and it has a very well-defined small genome. So we want to take that very well-defined small genome, chop it into smaller pieces. So instead of putting all 474 genes at a time, we want to make units of about 50 genes at a time. And we know we can probably express about 50 genes at a time. So we want to do that. We want to basically build it up by expressing 50 genes and then another 50 genes and so on, and then try to see what happens if we put together two synthetic cells expressing each 50 genes. And we end up with 100 genes. Is it still working? If it is, then we're going to keep adding. And that way, maybe we'll get to the point that Josiah was talking about, where you take a genome and try to express it. People have tried to do it in one go many times, and it always fails. The kind of a common belief why it always fails is because there is crap ton of transcriptional and translational regulation going on in cells that we don't understand. And so if you just take the genome, there are no natural transcriptional activators and all those signaling proteins that are telling each gene when to be expressed. And so you just end up with a big mess that doesn't really start ticking as a living system.
So just to start off, Kate, like how did you get into working on synthetic cells? Like what's your background?
I'm a chemist by training. I grew up doing organic chemistry. That was my undergrad. And then I worked on Origin of Life. I worked for about 10 years on astrobiology and origins problems.
Do you believe in aliens?
Not really. I mean,
no, come on.
They're out there, but they're not going to come. They're too far, and they're probably dead. So it's.
Oh, you think? What about like microbiological, maybe alien life? Do you think that's more likely that's out there, or
possible? The likelihood of bacterial type life existing somewhere in the universe is extremely high. I'm absolutely certain there is some out there. The likelihood of that life being ejected from their planet of the origin in a rock that's big enough that the core can preserve spores of life is somewhat lower, but still believable. The likelihood of the rock containing those aliens getting close enough to the solar system is extremely small. And then the likelihood of that actually getting in our hands is even smaller. As far as we know, we've only had, in the history of our scientific civilization, we only had 2 or 3 objects that were close enough that we could possibly grab it, that could potentially contain spores of life, assuming they were ejected from a planet that harbors life. So just most recently, the Oumuamua rock that was probably an flyover from out of the solar system was close enough that, if we were 50 years ahead in our space technology, we could probably go out and grab it. And then we could drill to the core of it, bring it home and analyze it, but we're not there yet.
So learning from your research, and a lot of scientists study the origin of life and say there was this RNA world and things like that. What's your opinion on maybe from your research on like how life started, because you intimately know like what's possible with cells being built, right? So like what is possible? What do you think it like started from, and how did it start, or you know?
I believe that under certain physical chemical conditions, life is inevitable. So if you give it enough time, and temperature, organics will start getting in trouble enough that it will evolve biochemistry. It's definitely happened gradually. It happened many, many times, most likely. I think there wasn't a single origin. That's why most people call this field of origins plural, because there were many instances of origin of something that we could sort of call life, and just one of them happened to win. I think the way it happens is single subsystems form first. So you start getting polymers, those polymers start acquiring catalytic activity, and slowly you build up complexity. And I honestly don't know how it happened. I can't give you an plausible scenario for the origin of our particular kind of life. We know it happened for sure, and I'm in the camp that says it happened on Earth. There's a lot of people that have made credible arguments for it happening elsewhere as well, but I mean, elsewhere like on Mars, and us being actually remnants of early Martian life. I think it actually happened on Earth, but we will probably never know. And I think it definitely happens slowly by PhD timescale, but fast by geological timescale. I mean, if you look at history of Earth, there was life on Earth as soon as we started having rock formation records. So there were only a few million years between the formation of crust and acquisition of water and when we can actually probe, and we know that there was life already back then. So the origin of life is a relatively fast process in the cosmological timescale.
So Idaho Bitcoin Group asks, what success do you see in the first synthetic cells? What is ultimate success, and what time frame do you think you're going to have with it?
So time frame is easy. I'm going to be up for tenure in 3 years, so that's my time frame. The first success will be, to me personally, a synthetic cell that can undergo a cell cycle, so it can grow and reproduce. We actually have every single component of that system known and experimentally proven already. That obviously devil is in the details. Putting it all together is the problem. We can have synthetic cells that grow, and we can replicate synthetic cells. We can replicate genome of synthetic cells and a compartment of synthetic cells, just never together. We can grow compartments of synthetic cells, and we can grow the guts of synthetic cells. We can make give it more of the protein expression system, but never together. So I think at this point, it's more of an engineering challenge. It's how do you put all of those components together? There's a lot of moving parts in the system. Honestly, I think that in about 10 years, we'll be able to say we have evolving, homostating synthetic cells.
Wow, that's pretty awesome. 10 years, ambitious. Here's a good question.
Yeah, go ahead.
Here's a good question by Anthony Lee. He says, are all synthetic cells organic?
Organic as in, the ones we work with, yes. We don't work in any silica, AI, oil, quartz, anything really. We work with honest proteins and lipids.
Cool. So do you think that all synthetic cells have to be self-replicating? No.
I didn't say self-replicating. I said replicating.
Replicating. Oh, interesting. So explain the difference.
Self-replicating is the cell just grows, and at some point decides to replicate. That's self-replicating. replicating means I grow them, and at some point I decide to replicate them, and I find a way to replicate them, either force them to undergo replication by adding some chemical to their membrane, or physically replicate them.
Yeah, that's really interesting. I never thought about that, because it is like we always think about cells in terms of like them deciding their own fate in every way, but like you could decide their fate, and they could be little robots like that. That's interesting.
Yeah, the power.
So Brunchable asks, it seems like the engine running these cells is made of cell-free protein transcription translation. Like, are these systems easy to use, work with? Do you purify the proteins from scratch? Do you just buy like extracts, or how does that work?
We make extract ourselves, and it's relatively easy. The extract we work with is a wholesale, most of the time, wholesale E. coli extract. So we basically grind up E. coli and do a little bit of purification. And we end up with a fraction that contains ribosomes, acyltransferases, and all of the chaperones and folding chaperones, transcription activators, ribosome assembly chaperone proteins, and then we add all the small molecules we think we need. We add tRNA, and we're done. And we add all the salts and a bunch of stuff. It's easy enough that biohacker labs have done cell-free prep extracts on their own before.
That's cool.
Yeah, when we ran the Fab Lab Bioacademy class, several labs have reproduced a protocol in their own biospaces, and
that would be awesome if you could like send that to us and we could post it up for the class, like a protocol for doing a simple experiment or something. That would be amazing.
So it's simple, but you need to have basically the kind of a base of the biolab setup space. So you need a centrifuge, you need either a -80 or dry ice, and you need vortex, set of pipettes, and you need to be able to grow bacteria. So
great. Yeah, no, we can figure it out. A lot of people have a lot of equipment that come with the class, so that's really cool. Here's an interesting question from Idaho Bitcoin Group. Do you think about like the lifespan of synthetic cells? You know, because E. coli, how long does an E. coli cell live? It's not that long, right? So like, what would be the lifespan of synthetic cell? Would it be like really long or short, or like what would it be?
So break it into two questions. E. coli doesn't really have a lifespan, because unless you kill them, they're not going to die. They're just going to divide. So E. coli cells never die of old age. If they die, it's an accident, disaster, antibiotic addition, or bleach. They never just shrivel and die on their own. They will just keep dividing. If you don't kill them, they'll keep dividing. And I forgot, there is some relatively small number of divisions after which they reach the mass of Earth. It's crazy how those bugs divide. So yeah, they never just die. So there is no such thing as lifespan of an E. coli cell, because they don't just sit there waiting to grow old. They will always keep dividing. Synthetic cells, on the other hand, they do sit and wait to grow old, and they do grow old. Most of the time, the cause of death is they run out of nutrients or energy. If we keep feeding them, we can keep supplying them nutrients and energy. And in this case, the cause of death will truly be in old age. Their ribosomes will start degrading and just won't be as efficient at protein synthesis. And that usually happens at, depending on many factors, at 2 to 4 days. So that's about the lifespan of those guys, unless you provide them with fresh ribosomes or a way to make fresh ribosomes.
Is there an optimal amount of time you'd want them to live?
Yes, we usually do experiments for about 12 to 18 hours. The reason for that is because you can set it up in the afternoon and go home. The real reason for that is if you look at protein expression, this is about enough time to see the protein expression to plateau. If you ever look at protein expression curves, there is this ramp up time where you keep making more and more and more protein, and at some point protein degradation kind of evens out with the protein production and you see a plateau. And that for most protein expression systems happens within 8 to 14 to 18 hours. It's a wide range because it depends on many different conditions. But that's about how long we want to do it. If we do experiments where we are actually fighting for longevity, those experiments can go on for days. We keep feeding synthetic cells new food, we keep giving it energy, we keep giving it fresh ribosomes sometimes. And then it basically just keeps going.
Kate, Kathy High asks, what are some of the ethical issues you face going forward with your work? Like, are you eventually going to grow, like organs to a baby, to like, tiny kittens? Like, what do you face?
I'm definitely not going to grow tiny kittens because I'm allergic to cats, but I would grow puppies if I could. The thing about it is people tend to overestimate synthetic cell technologies. They literally, at this point, are soap bubbles that make proteins. We are so far from growing anything complex that just this week, someone got a paper into a Nature Baby journal on simple communication via small molecules between basically oil droplet synthetic cells. And a month ago, there was a paper in Science for communication between synthetic cells via small protein. So this is basically where the field is. Everyone gets extremely excited when you can get two soap bubbles to talk to each other by one small molecule. We're so far away from growing organs, babies, kittens, and even puppies that it is not a consideration at all. The true ethical considerations of this technology come from being able to engineer genetic circuits that are not viable in natural systems. So you can basically imagine, again, we're not there yet, but you can realistically imagine building a synthetic cell that can express a protein that would be too toxic to express in live cells. But because synthetic cell starts out dead and kind of doesn't care, you can express really toxic proteins. It's actually, this system is used in industrial scale right now for expression of proteins that are toxic to live cell hosts. So you could imagine engineering genetic circuits that would be really hard to engineer in live cells, engineer them in synthetic cells, and then they could potentially produce either pathogenic products or create gene circuits that will then cause via horizontal gene transfer become pathogenic. And obviously we as a field have to be aware of those applications. There always can be bad actors. Nobody that I know will ever do experiments like that, but as with any technology, there are dangers. And by being aware of it, I mean, we need to follow the technology, we need to know what's currently available, and we need to be on the lookout, like with any other biological, potentially bioterrorist dangers. We need to be on the lookout for what is needed to do those experiments and how can we control the pipeline. So for example, the screening system we have in place right now so people cannot order an anthrax protein toxin from IDT, that should, as the technology progresses, should be extended into synthetic cell proteins that could potentially create potentially dangerous circuits. Again, we're not there yet with the technology, but we are aware of it and we're kind of looking into it.
Yeah, no. So all this sounds so sci-fi to me. Like, what's your, doesn't it though? Are you a big sci-fi fan?
I am.
Yeah. So like, what is your, I know there's like the stuff you want to do and stuff like that, but like, what's your like sci-fi future? Like, if everything goes right in the next 40 years and you get all the money you want to do the most amazing things, like, what's that sci-fi future you dream of with synthetic cells?
For one, synthetic cells that are good enough at biomanufacturing that we can get away from oil. That's actually one of the things that people don't think much about. Is everyone freaks out that we're running out of fossil fuels, and the solution to that is put more solar panels on your roof. But that only solves one problem. That solves the problem of where you get the energy from. But if you look at our current chemical technologies and chemical industry, there is so much indispensable chemicals that we get from fossil fuels, from oil, and there is no replacement, no biomanufacturing route to replace those chemicals right now. Most of the time, because those products are just extremely toxic to make. And this is the problem that people don't think about when they talk about replacing fossil fuels. It's not just replacing the energy, it's replacing all of the chemicals that we get from fossil fuels right now. There is no viable route at this point to biomanufacture in live cells all of those chemicals. It's possible that if you take a cell that doesn't really give crap about what's made in it, it's essentially dead to begin with, you can program it to make all the toxic stuff you want, because I mean, we still need that toxic stuff. So that is one application that I would love to see. Another one is to grow potatoes on Mars. And in order to grow potatoes on Mars, you first have to transform Martian soil into something that can actually support the terrestrial type vegetation, and then you need to give just a little bit of boost to the atmosphere. And that will most likely be done with a heavily engineered synthetic cells, because natural cells are not very good at being polyextremophiles. With all due respect to Deinococcus and water bears, they're great organisms, but they wouldn't want to live on Mars. And most terrestrial organisms, no matter how polyextremophile they are, they will not particularly care about living on Mars. We've seen what happened to that poor little plant on the moon. They just were not designed to live anywhere but on Earth. So in order to terraform Mars enough to actually grow potatoes there, we need to first start with baby steps by producing some sort of a synthetic cell that can slowly jumpstart this terraforming process. And planetary protection folks can now come and eat me. I believe that should be done, and that there are some people that say we should leave Mars to Martians and not terraform it. But that's another kind of a sci-fi application. Yet another one is for me to be able to build alternative living systems. So again, as I said earlier, we only have one type of biological system right now. Everything came from terrestrial last universal common ancestor, and that's all we can study. I would love to be able to study organisms that are totally weird. Like, what happens if your ribosome is completely different? What happens if your DNA is made of a different sugar backbone? What happens if your stereochemistry is reversed? All of those really cool questions that we cannot study with natural cells, I would love to be able to study them with synthetic cells. Is that enough crazy application?
Well, that's like all the practical applications, right? Like, I don't know. What is some impractical, like, kind of like out there idea you've had about synthetic cells?
I don't know. One really crazy one is if you're able to couple synthetic cells to neurons and then couple an electronic receiver to a synthetic cell, you could truly have a electronic neural interface. But that's totally crazy because that's not going to work. But I like the idea.
Yeah, so Christopher Henningsen asks, so the biggest hurdle to getting viable synthetic cells, a question of getting the environment to get the DNA to express properly?
Mm-hmm.
So what's kind of like the big hurdle to like getting synthetic cells to be viable?
Gene expression control is definitely the biggest problem. So basically, there is this famous physicist, again, physicists, they seem to have the best quotes. There's this famous physicist, Stu Kaufman, who said life, basically the core thing of life is adaptation at the edge of chaos. So basically, life is constantly barely limping along. But because of that, we can explore all the space of possibilities. We can evolve, we can do crazy stuff. We as life, like, we can evolve a platypus, we can evolve a hamster, we can evolve slime mold. In order to do that, there is extremely complex gene expression control machinery. A lot of cells' energy goes into deciding who gets to go right now, like which gene gets to express at which level, when do they have to shut down. And we not only don't understand a natural mechanism for doing that, but we also have no idea how to replicate that mechanism in vitro. And this is a really exciting problem that also pays the bills, because most of the time when something goes wrong, when we get sick, that's a problem with gene expression. We're making too much of some protein, not enough of some protein, or a cell goes high wire and decides to just keep replicating, which basically means keep expressing wrong genes at the wrong time. So if we can understand that control of gene expression, then we'll be golden, both for synthetic cells and for curing all diseases. That's obviously oversimplification, but it's a very important problem. And in order to build an actual viable synthetic cell that we can all agree we can call life, we need to be able to figure out this control of gene expression. Because right now I can put 100 genes into my synthetic cell. Heck, I can put 400 genes into my synthetic cell. I can make a whole Syn 3.0, the synthetic venter cell genome. I can put it into my synthetic cell and wait till I grow old and die. Nothing's going to happen. The genes are not going to start expressing correctly. And we don't know yet. We don't yet know why. So that is the biggest problem that we're trying to figure out right now.
So you went, you got your PhD, you know, now you're a professor at a university and all this stuff. So a lot of the people who are taking this class probably won't go to graduate school or
Good, don't.
So my question is, you know, like, you chose this path to go through, and like, why do you chose this path? What do you think are the benefits of the path you've chosen? What do you think are the benefits maybe of people not being down this path, and things like that?
If you're smart, you should not be on this path. Getting a faculty job is insanely hard. And when I decided I want to be a PI, I was a dumbass undergrad and I didn't know any better. I just had this idea that you're going to run a lab, it's going to be all unicorn farts and wonderful. It's this wonderful life. I had no idea how hard it is and how incredibly lucky you have to be able to run your own lab in academia. It's not as much due to being smart and hardworking, because there's a lot of smart and hardworking people that just don't get lucky. There's a lot of luck involved. There is luck at every step. If your PhD goes well, that's if your postdoc goes well, if you're lucky at the time you're looking for jobs, and also if you end up wanting to have a partner, then you end up with a two-body problem that again takes twice as much luck to solve that problem. So I would honestly not recommend people to go down that path. I think my job is amazing and I really can't imagine doing anything else right now. I really enjoy it, but I also recognize how incredibly lucky at many different stages I was to get where I am. It's not something that I can recommend with straight face to anyone. Maybe grad school, if you go into grad school with a strong focus on going into industry, that makes much more sense. If you don't go into grad school, more power to you. You're not going to waste the best years of your life sitting down and pipetting. I think there's a lot of amazing jobs in science, but there is many more people that think those jobs are cool, which is why I think we have an overproduction of PhDs right now and overproduction of postdocs. And most of those people, when they go into academia, they think, oh, I'm going to be a professor. And then by no fault of their own, they end up not being able to be a professor, and they just end up doing jobs that are either below their qualifications, or they could have been doing it years ago without all the training they spent a lot of time getting. So I think that generally biotechnology and synthetic biology sector is one of the fastest growing right now, and that's true. And it does need a lot of smart people, but being an academic should not be the pathway that most people choose when they think they choose when they go to grad school. I think if you want to go to grad school, the only smart way of doing it is to think, okay, I will go to grad school and then I will try to go make money in the industry. If you happen to stumble upon a lot of lucky coincidences during your postdoc, if it looks like everything's going well, you might start considering going on a faculty job market if you can take enough rejections and you're happy to live through that. But nobody should go into grad school thinking I'm going to be a PI. That's just a recipe for a disaster, for being frustrated and not really being happy. And I wish someone told me that when I started, but nobody did.
Mr. Smith.
Kate, Mr. Smith asked, do you believe that a capable, intelligent person like Mr. Smith could manufacture a synthetic cell with only an online education and no college/lab experience?
Sure. The no college, that's definitely true. I mean, you don't need to go to college to be able to read papers. No lab experience, that's a little tricky. I think if you set out on the quest to make a synthetic cell with no lab experience, by the time you actually get there, you will have enough lab experience because you're going to go through so many failures of your experiments that will give you an amazing lab experience. So it's always a learning process. And there is a lot of tricky experimental steps that a person coming from the street with no knowledge of how to pipe it will not be able to do it. But if you're patient and you just say, okay, that's my end goal, but I'm going to build up to it by doing certain experiments in the correct order, then sure, why not?
So, Kate, a lot of these people are, some of them are experienced, but some of them are just getting started in genetic engineering and things like that. So, like, you're super inspiring, but, like, what inspires you? What inspires you, to work on this stuff so hard every day? Because a lot of these people, it takes a long time, right? You've been studying this stuff for probably 15 years or something. So what inspires you to, like, keep you going every day, and, like, keep working on this stuff?
A paycheck is a good motivation. That's my job. But honestly, I think in current science, I know that's going to sound super cheesy, but there are really only two problems that I find infinitely fascinating and worth spending your life working on. One is solving the brain, figuring out how human brain computes what makes us who we are on the biological level. And the other one is, how do we engineer biology? How do we actually understand biological systems enough to engineer them from scratch? And I tried my hand at the neurobiology and solving the brain one. I didn't quite care for mouse work, so I ended up doing synthetic cell work. It's really cool problem because when you think about it, building cells from scratch is like dealing with every single biological problem all at once. You need to figure out every system, every subsystem of a living cell. You need to figure out how DNA gets replicated, how transcription works, how translation works, how membranes gets made, and everything in the middle. And that's just kind of incredibly fascinating to me because it helps you truly understand what is a living system. Not just spend your entire life studying this one pathway to lead to a disease. As valuable as those research programs are, I kind of like to play and I like to think. I'm kind of brain scattered, so I can't focus on one thing for too long. That's why I decided to just work on trying to make the entire cell, because then if I get tired of one problem, there is 20 different problems waiting around the corner to think about. And that's just kind of a cool life, is you can think about those things.
I like it. So it's just like basically you found a problem that fascinated you, a question that fascinated you, and you just decided to go after it and try to solve it, even if it was super complex and difficult and all these things. I think that's a really good, a really good way to think about it and do it, is like find a question that just interests you, even if it might seem like you're not capable of solving it right now, because along the way you learn so much and you keep going and you keep moving towards something. You keep pushing the field towards something, and when it does happen, it's just the coolest thing and the coolest feeling when you do solve something. That's awesome.
It really is. I I know that sounds awfully cheesy because that's exactly what the children's book will tell you a scientist does. But it's fun and I like doing it.
Yeah, question, Kate. So, this is not a measurement of like how apt you are, but how many times have you like failed throughout like your whole entire scientific career in terms of like, experiments, or like just like you get a project and you're like, this fucking sucks. I can't like do it anymore. Like how often do you experience failure to success?
So it would be actually easier to count successes than failures, because there's seriously, I mean, the research is mostly failures. Once you succeed on an experiment, you repeat it a few more times and you move on to look for new failures. Once you get a paper published, you move on to get 10 more papers rejected. Same with grant applications. Once you get something funded, you move on to write more grants that will inevitably get rejected. This whole job is about rejections, failures and rejections. And I guess that's why what I think separates good scientists from bad, not how hard you work or how smart you are, but how do you deal with crap? Like, how do you deal with your experiments failing? How do you deal with people you rely on failing? How do you deal with your work being criticized beyond recognition? Like when you get paper or grant reviews, they tell you you suck, you know nothing, you're good for nothing, this is completely stupid idea. And how quickly can you get over that? And then when you do experiments, you spend a few days working really hard and then you end up with nothing, not even negative result. It's just a clear failure. What do? Do you sit down and cry, or do you just kind of shrug and move on?
I usually cry and eat French fries.
Yeah, I actually prefer ice cream, but the similar strategy. So yeah, there's more rejection and failures than successes, which to me honestly kind of makes the successes worth more. But I know for a lot of people, the successes and failures count about the same, and because there is much more failures, the job ends up sucking. So I can't say how I deal with it. I just kind of accept that that's part of the job, and such is life. You move on. But definitely more failures than successes.
Yeah, that's like the most pinnacle thing about being a scientist, right?
Yeah.
We're going to about wrap this Google Hangouts up. And I'm kind of looking through people's questions if they have to ask you. Peter Gill asks, what would you like in someone that wants to join your lab? What do you look for in someone that wants to join your lab?
Good hands, good manual skills, and ability to learn new stuff relatively quickly. We don't expect people to come knowing any specific techniques. We train them in everything. So we're just looking for someone who's genuinely interested in the field. I don't want people joining my lab just because they need a lab. I want them to actually ask questions about our field. And that's pretty much all we need.
All right. That sounds good. So we're running a class with a bunch of people that have never experienced even genetic engineering or pipetting or any lab bench work. What kind of advice would you give to these people that are interested in getting in our sort of fields of like synthetic biology or genetic engineering or things like that?
I would say go out and start playing. There's so much simple experiments that can be done with almost no prior knowledge. And once you start doing them, it's like every other skill. You can't just go into the lab and do an experiment that's going to end up in a science paper. You go into the lab and grow bacteria. And if you're lucky, after about a week, you learn how to grow bacteria. And so basically baby steps. Always wear gloves. Kill your bacteria if you transformed them with antibiotic-resistant plasmid. That's a really important one. Do not pour your antibiotic-resistant bacteria down the drain. We're all screwed when you do that. So really, please don't. And that's pretty much it. I think a lot of this work is actually fun. And if your experiments don't work, that's kind of fun too, because you ended up getting to set it up and maybe you learned something, or you have no idea why it failed, so you just repeat it. And it fails again, but it's still fun. So I really think people shouldn't be intimidated by it. It's not like you have to be a scientist to be able to do simple synthetic biology experiments. You can just play with it in your spare time, and you do not have to be a scientist to do that.
Right. That's some really good advice. Yeah, I mean, that's why pretty much all of us got into it. We're just like, it's so intimidating, but I really want to learn about it. So like, how can I do such a thing? All right, cool. Yeah, thanks so much for hanging out with us.